So the first real major chemotherapy drug came out of the Second World War. There was a raid, an air raid on a naval base in Bari, in Italy, where they were storing mustard gas. And the mustard gas exploded and exposed a whole bunch of people to something. Autopsy studies revealed profound lymphoid and myeloid suppression, and that was also confirmed in survivors. Goodman and Gilman at Yale University then characterized this. They identified a compound called nitrogen mustard, killed lymphoid tissue, can it be used as a treatment for lymphoma? And bingo, it did just that, and the first chemotherapeutic agent was identified. It wasn't through empirical research. Faber, about 65 years ago, an Australian, found that giving folic acid to people with acute leukemia, kids with acute leukemia, actually made the leukemia worse. So, in a real empirical way, he and his colleagues developed folate analogs, which would block folate production in the body. He knew nothing about DNA. This was 1947. But he knew that if he blocked the production of folate, he might get an effect. And he did. And it was one of the first treatments for acute lymphocytic leukemia in children. And it worked. It gets better, actually. I love this stuff, because it's not just history, but it's serendipitous. Rosenberg at Michigan State University in the 60s was doing experiments to try and find out whether an electric current would kill bacteria. He had an experiment set up in his lab, and he had this culture medium, and he had electrodes on each side to produce the electric current. And he found that after 2 hours of applying electric current, bacteria died. And he said the electric current works. Terrific. Further analysis showed it wasn't electric current at all. It was a compound called cis-diamine dichloroplatinum. He said if it works in bacteria, it will work in cancer cells. And it did. And it is now one of the standard chemotherapy kind of drugs that's used across a very broad spectrum of activity in oncology. Another one was a Canadian bent, and for those who don't know the story, it's actually delightful. Clark Noble, working with Banting, Best, and Killer at that place called Toronto. I'm sorry, say something. 25 leaves from a periwinkle plant called Vinca rosea, widely used in Jamaica to treat diabetes. Well, Clark Noble didn't have time to investigate it, so he sent it to his brother, Robert Noble, at the University of Western Ontario. He tried it out to see whether he could treat diabetic mice with this compound. It didn't work, didn't budge in terms of blood glucose levels. But it sure as heck knocked the blood counts down and caused, lymphoid changes. And from that, the Vinca alkaloid was born. And the first patient treated with the Vinca alkaloid was at the Prince Martin Hospital, by Harold Warwick, and used today in Kristen and Blaston. It's actually a Canadian product, huge contribution. Taxol, for those of you who don't know, Taxol is a drug with terrific use in oncology. Came out of the NCI-US initiative in 1960, where they commissioned the US Department of Agriculture botanists to collect samples from 1,000 plant species a year. In 1962, Blackley collected bark from a Pacific yew tree. The extract was found to be cytotoxic, and Taxol was born. I'm sorry to disappoint those of you who like empirical research and targeted stuff, but these are realities that have occurred over time. Tamoxifen is the first personalized, what you call personalized medicine drug, I think, where the drug is used if you have estrogen receptors on your cell surface. From there we have the trailblazing kind of new generation of what I call personalized medicine drugs, where you can predict effect and you can give you a good idea whether the drugs are going to work or not. HER2 new trastuzumab, which is an antibody directed against HER2 new receptors, terrific drug. And the same thing with imatinib, which is used to treat chronic myeloid leukemia, and that is an antibody against BRCA protein. Great drugs. In fact, imatinib had the most rapid approval process in the history of the FDA, and it followed phase 2 studies because it worked so well. It was a pretty standard dunk kind of technology, and I think that's the kind of world that we would like to live in if we can. So I want to go to the last port of call. I want to talk about personalized medicine's clinical research implications. I'm almost done. How much time do I have? Yeah. Oh, let's relax. So this is where I want to talk about clinical predictive testing. The most important thing about clinical predictive testing, from my standpoint as a evidence-based methodologist, is how does a new predictive test compare in terms of accuracy, budget impact, and cost effectiveness with alternatives? You have to answer that question to get any space in the health system. I would throw up a question of accuracy statistics in terms of predictive testing. We have summary accuracy statistics that tell you area under the curve for an ROC curve, which combines sensitivity and specificity and give you an idea of the combined accuracy of the test. I would encourage you to always look at the sensitivity and the specificity separately, because if you're going to make a clinical decision based on both of these without understanding what you're doing, you could be exposing patients to unnecessary risk. So if the false positive rate is very high, or if the false negative rate is very low, then you need to know that instead of just looking at summary statistics, because either one of those is going to disadvantage patients. And I encourage you, whenever you're seeing summary statistics, to ask for the disaggregation of the sensitivity and the specificity, because they can have significant implications in policy decision making. Not just that, but patients want to know what they're letting themselves in for. And it's something that I have found increasingly is everyone is enamored by genomic predictive testing, and quite rightly so. I think it's a terrific development. I mean, who's not going to get excited about some of the stuff that's going on at the moment with protein kinase inhibitors, mTOR inhibitors, all that really good stuff. But don't get into it without doing a comparative effectiveness analysis to know exactly what you're on about. We did an analysis on Oncotype DX, which allows you to determine which patients are at highest risk for stage 1 breast cancer, who should get chemotherapy, who should not get chemotherapy. It looks great. Has two problems, by the way. The one is the evidence on which Oncotype DX evidence-based analysis is based is low-quality evidence. So we had a health economist take us through this huge, wonderful, complex, amazing economic model that said that it's cost-effective no matter who you use it in stage 1 breast cancer. But the problem is that everything he put in his model was based on low-quality evidence. So now what are you going to do with that stuff? I'll tell you what we did in a minute. So when it comes to Oncotype DX, compare it to adjuvant online, which is another risk stratification mechanism which looks at all the known prognostic variables for breast cancer. It's free. You can get it off the internet almost. And a lot of oncologists, breast oncologists, continue to use it. So know the world you live in and know about comparative effectiveness. When it comes to pharmacogenomic tests, some of the policy considerations, who pays for the test? You know what, I'm not even sure that it's a problem. I can't see why this is an issue for policy decision makers. Who pays for estrogen receptor analysis in breast cancer? Who pays for HER2/neu expression in breast cancer? I don't think it's a policy issue. I really can't see a drug being developed based on a target where the payer is going to pay for the drug and not the target. And our experience so far, certainly in Ontario, has been exactly that, where industry is not paying for the actual diagnostic test. Government has said, the Ministry of Health and the drug branch has said, well, we will kind of pick it up. We'll go with that because it's coupled. So, I think a lot's been said about this. I'm not sure that in a policy arena it's going to have much of a problem. But who oversees the QA component of the test is a really interesting issue. Because if the test is bundled with the drug, like the FDA have recently done with BRAF and an anti-BRAF drug, which we'll come to in a second, then Health Canada says it will oversee the quality assurance of the test. But if it's being done in a lab in a hospital, like EGFR, for example, receptor, the government kind of steps back and says, you guys are in control. We don't really think it's important for us to do that. It's a policy issue in that there is no policy. As of a year ago, could have changed in the last year. And then the last policy consideration is, we all know we're getting into the specific area of drug testing and targets. And we all think that we are going to diffuse the drug according to the match with the target. But what happens when you have, this new drug, berufimab, I forgot what it's called anyway, this anti-BRAF drug for melanoma, which has a 65% response rate. Now, I'm not sure as an oncologist that I like overall response rates, but anyway, 65% response rate compared to standard chemotherapy for melanoma, which runs at about 5–10%. So can we anticipate off-label creep of the use of this new drug for people who test negative? And it's quite possible that when you're sitting in a room with a patient and everything is failed and you're trying a drug and it's 5% response rate and things, you're up against the wall, would you try a drug that has a 65% response rate even though it's targeted? Can it do more than the 5 or 10% that conventional chemotherapy is? So off-label creep, which goes beyond the test, is something you need to think about. And I would say that in evaluating pharmacogenomics, I don't see any real problem with this at all. Use the standard methodologies for assessing evidence. You may have some additional considerations, and I'll put them up on the slide, which I won't go into. But there are certain studies where you can collect archival material, and then you can go back and you can apply it to randomized controlled trials, so-called retrospective, prospective study design. And Richard Simon's written extensively on this, and it does have validity. So there's some neat kind of twists to it in terms of methodology, but I don't see it as a major issue, and in terms of evidence development. And then consider coverage with evidence development if there's residual uncertainty. So this is what we've done in our unit in terms of pharmacogenomic testing. And you'll see in the right column is what's been approved. Lung cancer, we look at EGFR genotyping to predict response to lockdown and gefitinib. And I have to tell you that the data on gefitinib in particular was really compelling. 71% response rate if you have the EGFR mutation, 1% response rate if you don't. Tell me about specificity, for goodness sake. And this is approved, and although Health Canada doesn't insist that you do the EGFR receptor in using gefitinib or relotinib, we're using it certainly for gefitinib first line in Ontario. In colorectal cancer, KRAS genotyping to predict response to cetuximab and panitumumab. Thought it really works. It's statistically significant, but you look at the response rates and you see that the relapse-free survival, the difference is measured in like 3 months, 5 months. So I don't know how wonderful these drugs are, and we need to have a look at not statistical significance, but clinical significance in treating with these new drugs.